Schistosoma mansoni is a water-borne parasite of humans, and belongs to the group of blood flukes. Schistosoma. The adult lives in the blood vessels, mesenteric veins, near the human intestine. It causes intestinal schistosomiasis, similar to S. japonicum, S. mekongi, S. guineensis, and S. intercalatum. Clinical symptoms are caused by the eggs. As the leading cause of schistosomiasis in the world, it is the most prevalent parasite in humans. It is classified as a neglected tropical disease. As of 2016, 206.5 million people have schistosomiasis, and S. mansoni is the major parasite. It is found in Africa, the Middle East, the Caribbean, Brazil, Venezuela and Suriname. Unlike other flukes, trematodes, in which sexes are not separate, monoecious, schistosomes are unique in that adults are divided into males and females, thus, dioecious. However, the two adults live in permanent partnership, a condition called copula. For this, they are considered as hermaphrodites. The life cycle of schistosomes includes two hosts: humans as definitive hosts, where the parasite undergoes sexual reproduction, and snails as intermediate hosts, where a series of asexual reproductive takes place. S. mansoni is transmitted through water, where freshwater snails of the genus Biomphalaria act as intermediate hosts. The larvae are able to live in water and infect the host by directly penetrating the skin. Prevention of infection is done by improved sanitation and killing the snails. Infection is treated with praziquantel. S. mansoni was first noted by Theodore Maximilian Bilhatz in Egypt in 1851. While discovering S. hematobium, Sir Patrick Manson identified it as a unique species in 1902. Louis Weston Rasambon gave the name Schistosoma mansoni in 1907 in honor of Manson. Structure: adult schistosomes. Unlike other trematodes, are long and cylindrical worms. The male S. mansoni is approximately 1 cm long, 0.6 to 1.1 cm, and is 0.1 cm wide. It is white, and it has a funnel-shaped oral sucker at its anterior end followed by a second pediculated ventral sucker. The external part of the worm is composed of a double bilayer, which is continuously renewed as the outer layer, known as the membranocalyx, and is shed continuously. The tegument bears a large number of small tubercules. The suckers have small thorns in their inner part as well as in the buttons around them. The male genital apparatus is composed of 6 to 9 testicular masses, situated dorsally. There is one deferent canal beginning at each testicle, which is connected to a single deferent that dilates into a reservatory. The seminal vesicle, located at the beginning of the gynecophoric canal. The copula happens through the coaptation of the male and female genital orifices. The female has a cylindrical body, longer and thinner than the male's, 1.2 to 1.6 cm long by 0.016 cm wide. It has the general appearance of a roundworm. The female parasite is darker, and it looks gray. The darker color is due to the presence of a pigment, hemozoin, in its digestive tube. This pigment is derived from the digestion of blood. The ovary is elongated and slightly lobulated and is located on the anterior half of the body. A short oviduct conducts to the ootype, which continues with a uterine tube. In this tube it is possible to find 1 to 2 eggs, rarely 3 to 4. But only 1 egg is observed in the ootype at any one time. The genital pore opens ventrally. The posterior 2/3 of the body contain the vitellogenic glands and their winding canal, which unites with the oviduct a little before it reaches the ootype. The digestive tube begins at the anterior extremity of the worm, at the bottom of the oral sucker. The digestive tube is composed of an esophagus, which divides in two branches, right and left, and that reunite in a single cecum. The intestines end blindly, meaning that there is no anus. Egg. The eggs are oval-shaped, measuring 115 to 175 micrometers long and 45 to 47 micrometers wide, and approximately 150 micrometers diameter on average. They have pointed spines towards the broader base on one side, i.e., lateral spines. This is an important diagnostic tool because co-infection with S. hematobium, having a terminal spinned eggs, is common, and they are hard to distinguish. When the eggs are released into the water, a lot of them are immature and unfertilized so that they do not hatch. When the eggs are larger than 160 micrometers in diameter, they also fail to hatch. Larva. The miracidium, from the Greek word mu epsilon iota rho alpha kappa iota omicron nu, merakion, meaning youth, is pear-shaped, and gradually elongates as it ages. It measures about 136 micrometers long and 55 micrometers wide. The body is covered by a nucleate epidermal plate separated by epidermal ridges. The epidermal cells give off numerous hair-like cilia on the body surface. There are 17 to 22 epidermal cells. Epidermal plate is absent only at the extreme anterior called apical papilla, or terebratorium, which contains numerous sensory organelles. Its internal body is almost fully filled with glycogen particles and vesicles. The ceraria has a characteristic bifurcated tail, classically called furcae, Latin for fork. Hence, the name, derived from a Greek word kappa epsilon rho kappa omicron sigma, kirkos, meaning tail. The tail is highly flexible and its beating propels the ceric area in water. It is about 0.2 millimeters long and 47 micrometers wide, somewhat loosely attached to the main body. The body is pear-shaped and measures 0.24 millimeters in length and 0.1 millimeter in width. Its tegument is fully covered with spine. A conspicuous oral sucker is at the apex. As a non-feeding larva, there are no elaborate digestive organs. Only esophagus is distinct. There are 3 pairs of mucin glands connected laterally to the oral sucker at the region of the ventral sucker. Physiology. Feeding and nutrition. Developing Schistosoma mansoni worms that have infected their definitive hosts prior to the sexual pairing of males and females require a nutrient source in order to properly develop from Cherkeriae to adults. The developing parasites lice host red blood cells to gain access to nutrients. The hemoglobin and amino acids the blood cells contain can be used by the worm to form proteins, while hemoglobin is digested intracellularly, initiated by salivary gland enzymes. Iron waste products cannot be used by the worms, and are typically discarded via regurgitation. Cashau et al. 1995, tested the effect of temperature and pH on the ability of developing S. mansoni to lyse red blood cells. The researchers found that the parasites were best able to destroy red blood cells for their nutrients at a pH of 5.1 and a temperature of 37 degrees Celsius. Locomotion. S. mansoni is locomotive in primarily 2 stages of its life cycle. As Cherkeriae swimming freely through a body of fresh water to locate the epidermis of their human hosts, and as developing and fully fledged adults, migrating throughout their primary host upon infection. Cherkeriae are attracted to the presence of fatty acids on the skin of their definitive host, and the parasite responds to changes in light and temperature in their freshwater medium to navigate towards the skin. Resurreissau et al. 2015 tested the roles of various protein kinases in the ability of the parasite to navigate its medium and locate a penetrable host surface. Extracellular signal-regulated kinase and protein kinase C both respond to changes in medium temperature and light levels, and the stimulation of p38 mitogen-activated protein kinase, associated with recognition of parasite host surface, results in a glandular secretion that deteriorates the host epidermis and allows the parasite to burrow into its host. The parasite's nervous system contains bilobed ganglia and several nerve cords which splay out to every surface of the body. Serotonin is a transmitter distributed widely throughout the nervous system and plays an important role in nervous reception and stimulating mobility. Life cycle. Intermediate host. After the eggs of the human-dwelling parasite are emitted in the feces and into the water, the ripe miracidium hatches out of the egg. The hatching happens in response to temperature, light and dilution of feces with water. The miracidium searches for a suitable freshwater snail belonging to the genus Biomphalaria. In South America, the principal intermediate host is Biomphalaria glabrata, while B. stramina and B. tenagophila are less common. A land snail, Achatina felica, was reported in 2010 to act as a host in Venezuela. In Africa, B. glabrata, B. pfeifferi, B. choanomphala and B. sudanica act as the hosts. But in Egypt, the main snail host is B. alexandrina. Myracidia directly penetrate the soft tissue of snail. Inside the snail, they lose their cilia and develop into mother sporocysts. The sporocysts rapidly multiply by asexual reproduction, each forming numerous daughter sporocysts. The daughter sporocysts move to the liver and gonads of the snail, where they undergo further growth. Within 2 to 4 weeks, they undergo metamorphosis and give rise to fork-tailed cercariae. Stimulated by light, hundreds of cercariae penetrate out of the snail into water. Definitive host. The cercaria emerge from the snail during daylight and they propel themselves in water with the aid of their bifurcated tail, actively seeking out their final host. In water, they can live for up to 12 hours, and their maximum infectivity is between 1 and 9 hours after emergence. When they recognize human skin, they penetrate it within a very short time. This occurs in 3 stages. An initial attachment to the skin, followed by the creeping over the skin searching for a suitable penetration site, often a hair follicle, and finally penetration of the skin into the epidermis using cytolytic secretions from the cercarial postacetabulum, then preacetabulum glands. On penetration, the head of the cercariae transforms into an endoparasitic larva, the schistosomule. Each schistosomule spends a few days in the skin and then enters the circulation starting at the dermal lymphatics and venules. Here, they feed on blood, regurgitating the HAEM as hemozoin. The schistosomule migrates to the lungs 5 to 7 days post-penetration, and then moves via circulation through the left side of the heart to the hepatoportal circulation, greater than 15 days, where, if it meets a partner of the opposite sex, it develops into a sexually mature adult and the pair migrate to the mesenteric veins. Such pairings are monogamous. Male schistosomes undergo normal maturation and morphological development in the presence or absence of a female. Although behavioral, physiological and antigenic differences between males from single sex, as opposed to bisex, infections have been reported. On the other hand, female schistosomes do not mature without a male. Female schistosomes from single-sex infections are underdeveloped and exhibit an immature reproductive system. Although the maturation of the female worm seems to be dependent on the presence of the mature male, the stimuli for female growth and for reproductive development seem to be independent from each other. The adult female worm resides within the adult male worm's gynecophoric canal, which is a modification of the ventral surface of the male, forming a groove. The paired worms move against the flow of blood to their final niche in the mesenteric circulation, where they begin egg production. Greater than 32 days. The mansoni parasites are found predominantly in the small inferior mesenteric blood vessels surrounding the large intestine and cecal region of the host. Each female lays approximately 300 eggs a day, one egg every 4.8 minutes, which are deposited on the endothelial lining of the venous capillary walls. Most of the body mass of female schistosomes is devoted to the reproductive system. The female converts the equivalent of almost her own body dry weight into eggs each day. The eggs move into the lumen of the host's intestines and are released into the environment with the feces. Genome. Schistosoma mansoni has 8 pairs of chromosomes. 2n equals 16. 7 autosomal pairs and 1 sex pair. The female schistosome is heterogametic, or ZW, and the male is homogametic, or ZZ. Sex is determined in the zygote by a chromosomal mechanism. The genome is approximately 270 megabytes with a GC content of 34%. 4 to 8% highly repetitive sequence, 32 to 36% middle repetitive sequence, and 60% single copy sequence. Numerous highly and moderately repetitive elements are identified, with at least 30% repetitive DNA. Chromosomes range in size from 18 to 73 megabytes and can be distinguished by size, shape, and C-banding. In 2000, the first back library of schistosome was constructed. In June 2003, a approximately 5x whole genome shotgun sequencing project was initiated at the Sanger Institute. Also in 2003, 163,000 ESTs, express sequence tags, were generated by a consortium headed by the University of São Paulo from 6 selected developmental stages of this parasite, resulting in 31000 assembled sequences and an estimated 92% of the 14,000 gene complement. In 2009, the genomes of both S. mansoni and S. japonicum were published, with each describing 11,809 and 13,469 genes, respectively. S. mansoni genome has increased protease families and deficiencies in lipid anabolism, which are attributed its parasitic adaptation. Protease included the invadolysin, host penetration, and cathepsin, blood feeding, gene families. In 2012, an improved version of the S. mansoni genome was published, which consisted of only 885 scaffolds and more than 81% of the bases organized into chromosomes. Pathology. Schistosome eggs, which may become lodged within the host tissues, are the major cause of pathology in schistosomiasis. Some of the deposited eggs reach the outside environment by passing through the wall of the intestine. The rest are swept into the circulation and are filtered out in the periportal tracts of the liver, resulting in periportal fibrosis. Onset of egg laying in humans is sometimes associated with an onset of fever, Katayama fever. This acute schistosomiasis is not, however, as important as the chronic forms of the disease. For S. mansoni and S. japonicum, these are intestinal and hepatic schistosomiasis. associated with formation of granulomas around trapped eggs lodged in the intestinal wall or in the liver, respectively. The hepatic form of the disease is the most important. Granulomas here giving rise to fibrosis of the liver and hepatosplenomegaly in severe cases. Symptoms and signs depend on the number and location of eggs trapped in the tissues. Initially, the inflammatory reaction is readily reversible. In the latter stages of the disease, the pathology is associated with collagen deposition and fibrosis, resulting in organ damage that may be only partially reversible. Granuloma formation is initiated by antigens secreted by the myristidium through microscopic pores within the rigid eggshell, and the immune response to granuloma, rather than the direct action of egg antigens, causes the symptoms. The granulomas formed around the eggs impair blood flow in the liver and, as a consequence, induce portal hypertension. With time, collateral circulation is formed and the eggs disseminate into the lungs, where they cause more granulomas, pulmonary arteritis and, later, cor pulmonale. A contributory factor to portal hypertension is Simmers fibrosis, which develops around branches of the portal veins. This fibrosis occurs only many years after the infection and is presumed to be caused in part by soluble egg antigens in various immune cells that react to them. Recent research has shown that granuloma size is consistent with levels of IL-13, which plays a prominent role in granuloma formation and granuloma size. IL-13 receptor alpha-2, IL-13 alpha-2, binds IL-13 with high affinity and blocks the effects of IL-13. Thus, this receptor is essential in preventing the progression of schistosomiasis from the acute to the chronic and deadly stage of disease. Synthetic IL-13 alpha-2 given to mice has resulted in significant decreases in granuloma size, implicating IL-13 alpha-2 as an important target in schistosomiasis. Mansoni infection often occurs alongside those of viral hepatitis, either hepatitis B virus, HBV, or hepatitis C virus, HCV. This is due to high prevalence of schistosomiasis in areas where chronic viral hepatitis is prevalent. One important factor was the development of large reservoir of infection due to extensive schistosomiasis control programs that used intravenously administered tartar emetics since the 1960s. Co-infection is known to cause earlier liver deterioration and more severe illness. Evasion of host immunity. Adult and larval worms migrate through the host's blood circulation, avoiding the host's immune system. The worms have many tools that help in this evasion, including the tegument, antioxidant proteins, and defenses against host membrane attack complex, MAC. The tegument coats the worm and acts as a physical barrier to host antibodies and complement. Host immune defenses are capable of producing superoxide, but these are counter-attacked by antioxidant proteins produced by the parasite. Schistosomes have 4 superoxide dismutases, and levels of these proteins increase as the schistosome grows. Antioxidant pathways were first recognized as choke points for schistosomes, and later extended to other trematodes and cestodes. Targeting of this pathway with different inhibitors of the central antioxidant enzyme thioredoxin, glutathione reductase, TGR, results in reduced viability of worms. Decay accelerating factor, DAV, protein is present on the parasite tegument and protects host cells by blocking formation of MAC. In addition, schistosomes have 6 homologs of human CD59 which are strong inhibitors of MAC. Diagnosis. The presence of S. mansoni is detected by microscopic examination of parasite eggs in stool. A staining method called cat-o-cat technique is used for stool examination. It involves methylene blue stained cellophane soaked in glycerine or glass slides. A bit costlier technique called formalin ether concentration technique, FECT, is often used in combination with a direct fecal smear for higher accuracy. Serological and immunological tests are also available. Antibodies and antigens can be detected in the blood using ELISA to identify infection. Adult worm antigens can be detected by indirect hemagglutination assays, IHAs. Polymerase chain reaction, PCR, is also used for detecting the parasite DNA. Circulating cathodic antigen, CCA, in urine can be tested with lateral flow immune chromatographic reagent strip and point-of-care POC tests. Treatment. The standard drug for mansoni infection is praziquantel at a dose of 40 mg per kilogram. Oxamniquine is also used. Epidemiology. As of WHO report in 2016, 206.5 million people worldwide are having schistosomiasis due to different species of schistosoma. More than 88 million people were given medical treatment. Mansoni is the major species. It is endemic in 55 countries and most prevalent in Africa, the Middle East, the Caribbean, Brazil, Venezuela and Suriname. About 80–85% of schistosomiasis is found in sub-Saharan Africa, where hematobium, intercalatum and mansoni are endemic. Approximately 393 million Africans are at risk of infection from mansoni, of which about 55 million are infected at any moment. Annual death due to mansoni is about 130,000. The prevalence rate in different countries of Africa are 73.9% in northern Ethiopia, 37.9% in western Ethiopia, 56% in Nigeria, 60.5% in Kenya, 64.3% in Tanzania, 19.8% in Ghana, and 53.8% in Côte d'Ivoire. In Egypt, 60% of the population in the northern and eastern parts of the Nile Delta and only 6% in the southern part are infected. Mansoni is commonly found in places with poor sanitation. Because of the parasite's fecal-oral transmission, bodies of water that contain human waste can be infectious. Water that contains large populations of the intermediate host snail species is more likely to cause infection. Young children living in these areas are at greatest risk because of their tendency to swim and bathe in Cercaria-infected waters longer than adults. Anyone traveling to the areas described above, and who is exposed to contaminated water, is at risk of schistosomiasis. History. The intermediate hosts Biomphalaria snails are estimated to originate in South America 95 to 110 million years ago, but the parasite's Schistosoma originated in Asia. In Africa, the progenitor species evolved into modern mansoni and hematobium around 2 to 5 million years ago. It reached Egypt via infected slaves and baboons from the land of Punt through migrations that occurred possibly as early as the V dynasty, c. 2494–2345 BCE. A German physician, Theodor Maximilian Bilhatz, was the first to discover the parasite in 1851. While working at Kasr-El-Aini Hospital, a medical school in Cairo, Bilhatz recovered them from autopsies of dead soldiers, and noticed two distinct parasites. He described one of them as Distemum hematobium, now hematobium, in 1852, but failed to identify the other. In one of his letters to his mentor Carl Theodor von Siebold, He mentioned some of the eggs were different in having terminal spines while some had lateral spines. Terminal spinned eggs are unique to S. hematobium, while lateral spines are found only in S. mansoni. Bill Hatz also noted that the adult flukes were different in anatomy and number of eggs they produced. He introduced the terms Bilhartsia and Bilhartziasis for the name of the infection in 1856. A German zoologist, David Friedrich Wineland, corrected the genus name to Schistosoma in 1858, and introduced the disease name as Schistosomiasis. The species distinction was first recognized by Patrick Manson at the London School of Hygiene and Tropical Medicine. Manson identified lateral spinned eggs in the feces of a colonial officer earlier posted to the West Indies, and concluded that there were two species of Schistosoma. An Italian-British physician, Louis Westerner Sambon, gave the new names Schistosomum hematobium and Schistosomum mansoni in 1907. The latter two honor Manson. Sambon only gave partial description using a male worm. In 1908, a Brazilian physician, Manuel Augusto Pirahá da Silva, gave a complete description of male and female worms, including the lateral spinned eggs. Pirahá da Silva obtained specimens from 3 necropsies and eggs from 20 stool examinations in Bahia. He gave the name S. americanum. The species identity was confirmed in 1907 by British parasitologist Robert Thomson Leeper, identifying the specific snail host and distinguishing the egg structure, thereby establishing the life cycle. Disease info at CDC. Taxonomy at ITIS report. Species profile at Animal Diversity Web. Profile at Wormbase.